We start now for the next session, technical session 8, on aquatic animal epidemiology, disease surveillance and reporting. I request the chairs of the session. We welcome Dr. Kenton Morgan Lloyd, Dr. Idya Karnasagar, please. We also welcome on the stage today's keynote speaker, Dr. Elman J. Peeler, lead presentation by Dr. Neeraj Sood. I request Dr. Neeraj to kindly come over the stage. We have the third presentation by the lead presenter, Dr. Alicia Gallardo Lagno. I request Dr. Alicia also to kindly come on the stage. Dr. Neeraj, please. So for this session, we have session coordinators, Dr. Chandra Bhushan and Dr. Elil Pravina. I request the coordinators to kindly coordinate the session.
A very good morning to all of you. Before we go into the session, it's my honor and privilege to welcome our chairperson, Dr. Kenton Morgan. And Dr. Kenton Morgan is a versatile personality who has done pioneering in veterinary epidemiologist and who has held the first chair of epidemiology at the UK Veterinary School in the year 1996. His research career includes diagnostic pathology and preventive medicine. He was a reader in the veterinary epidemiology from the year 1993 at the University of Bristol and became head of Department of Animal Health and Husbandry in the same year. He developed the largest epidemiology group in a UK veterinary school and trained over 30 postgraduate students. Dr. Morgan has introduced aquatic epidemiology in the year 1998, leading to his presidency of the International Society for Aquatic Animal Epidemiology from the year 2007 to 2012. His global contributions include a visiting professorship at the University of Nagasaki from the year 2013 to 2016, where he designed an intensive epidemiology course. After his retirement in the year 2016, he returned to the university, graduating with an MA in acting in the year 2019. Now, he divides his time between his passion for teaching epidemiology and that of professional teaching. I welcome you, sir. And now I request my colleague, Dr. Chandra Bhushan, to introduce the other chairperson.
Thank you, madam. It's my pleasure to introduce Dr. Idya Karnasagar, who is a very familiar to most of us. As you know, Dr. Karnasagar is a distinguished microbiologist and global authority on fish safety and quality. He held a prominent career in Indian academia, rising to professor and director of research and earning the ICR National Professorship. Internationally, he was a senior fisheries officer at the FAO of the UN in Rome, where he led initiatives on fish safety, contributed to Codex Alimentarius international food standards, and was pivotal in creating the WHO 5 keys for safer aquaculture products. He is currently a member of key WHO advisory groups on antimicrobials and foodborne diseases, and currently serves as a research advisor at Nite University. Thank you, and I request chairs for further proceedings. Thank you.
So I have the great honor and pleasure to introduce Professor Edmund Peeler, the keynote speaker in this session. Professor Edmund Peeler is a UK-based epidemiologist and principal scientist at CFAS with over 2 decades of leadership in aquatic animal health policy, disease surveillance, and risk analysis. He advised the UK government and EU on research priorities and served as Vice President of the UHA Aquatic Animal Health Standards Commission from 2015 to 21. With over 70 peer-reviewed publications, he continues to support international standard development and consults globally on aquaculture health systems, and received the UHA Meritorious Service Medal in 2024. So great pleasure to invite Edmund Peeler to give his keynote address.
So the first international meeting I attended after joining CFAS was DAA 5 in Australia in 2002. So it's an enormous pleasure and privilege to have the opportunity to talk to you today. And I'd like to thank Kuldeep and Melba and the other organizers of this meeting for the opportunity. So as you've heard, I work at CFAS, and as my colleague Andy has mentioned, we have a stall. So if you haven't managed to do so, please find the time today to talk to my colleagues about our WOA Collaborating Centre and our FAO AMR Reference Lab. So I will talk about the importance of early detection systems, and this is the rough outline of what I'm going to cover, but I'm going to very much focus on barriers to disease reporting and what we can do to improve it. As other speakers this week have mentioned, aquaculture will inevitably continue to grow to meet global demand. It means that we will farm in new regions with new species. Diseases will inevitably continue to emerge. The threat of transboundary diseases is likely only to increase as new trade patterns emerge. And we trade within the rules set by the SBS agreement of the WTO. And under those rules, if a country wants to protect its disease-free status, it needs to demonstrate freedom according to WOA standards. And indeed, under those standards, there's not just a legitimacy to erecting trade barriers, there is actually an obligation to do so. So I thought I'd start by a definition, taking the WOA definition of an early detection system. It is a system in which there is rapid recognition of suspicion of listed diseases, emerging diseases, and I think very importantly, elevated unexplained mortality. And also, it's not just recognition, but also the rapid communication of those events to the competent authority and an initiation of an investigation. And we can think about different components to the early detection system. I put a few of the most important ones there. And in this talk, I'm going to focus on farmer reporting. If we were looking at surveillance in wild populations, we would obviously have a very different set of surveillance system components. Okay, so why is early detection important? Okay, so sticking with the concept of disease freedom, as mentioned, if a country wishes to protect that disease-free status, it needs to demonstrate freedom to WOA standards, and they are encapsulated in basic biosecurity conditions. So most importantly, measures to prevent introduction, effectively trade barriers based on disease-free status, but as importantly, an early detection system. And WOA define these 5 criteria for an effective early detection system, and I will touch on these in a little bit more detail in a minute. But in a way, more importantly, early detection is crucial because we know that many diseases, when introduced or emerged, will spread rapidly. They have, in epidemiological terms, high reproductive value. And really nice to see that there's a presentation later today looking at the R0 value of tilapia lake virus. So early detection is critical to the success to a cost-effective response. There is only a narrow window of opportunity when it may be both economically feasible and technically possible to control and hopefully eradicate an emerging or introduced disease. And we know that disease outbreaks can have national level impacts. For those of us in the UK interested in animal health, the 19th of February 2001 is a significant date. It was actually the day I started work at CFAS, but somewhat more importantly, it was also the day on which foot and mouth disease virus was detected in pigs in an abattoir in the south of England. But by that date, the virus had already spread, and it resulted in an outbreak affecting 2,000 premises, the slaughter of over 6 million cattle and sheep, a cost to the UK economy of approximately £8 billion, and as importantly, a massive toll of human suffering. So, you know, we can view the foot and mouth disease outbreak as a failure of biosecurity, but it is also a failure of early detection, which could have resulted in a far smaller outbreak. And of course, we've heard today of examples, this week, of examples in the aquatic side. Obviously, white spot's a classic example, and we heard this morning from Victoria about the impact of its introduction in Saudi Arabia. You know, Ecuador is another standout example where the impact of white spot had economic consequences at a national level. Rahana mentioned this ISKNV outbreak in Ghana, you know, almost halving the level of tilapia production. So these pathogens can present existential threats to the aquaculture industry. Okay, so I'm briefly going to kind of digress a little bit into how we assess surveillance systems, because as mentioned, if you know, WOA require countries to assess their system if they want to meet their standards. But I think more importantly, I would argue that competent authorities should take time to assess their early detection systems to identify where weaknesses are, where investment is needed to improve the system. And as mentioned, as the quotation there states, if you can't measure it, it's difficult to manage it. So firstly and most simply, one can take the WOA criteria and use them as a checklist. So there needs to be awareness of farmers and others of listed diseases, and aquatic animal health professionals need to be trained in detection and reporting. Very importantly, there needs to be capacity of the system to rapidly investigate and respond to an outbreak. There needs to be diagnostic capacity and legal obligation to report suspicion of listed diseases, which presupposes that legislation lists diseases. And clearly, you know, taking number 3, the evidence has to be examples of disease investigations, examples of investigations into unexplained mortality. Sticking with WOA, you don't need to wait for an external audit to make use of the PVS tool. You could use this tool to assess any aspect of an aquatic animal health system. I think it's a really useful resource, and I would urge you to look at it if you're wanting to do an evaluation. Melba's already mentioned this paper, so, you know, excellent, again, a checklist, excellent 12-point checklist. I think it's really useful, especially if you're wanting to have a model to help support capacity building. And then staying with the FAO, there is the surveillance evaluation tool. This is very comprehensive, 96 indicators. It was obviously designed with terrestrial systems in mind, but it has successfully been applied to look at disease surveillance in fish farms in Spain. So the tools I've discussed so far are qualitative or semi-qualitative, which, to be honest, is perfectly adequate as far as WOA standards are concerned. But however, there can be advantages to taking a quantitative approach. Angus Cameron, Tony Martin, and others developed scenario tree modeling approach some years ago for this purpose, to assess quantitatively the likelihood that a surveillance system is going to detect a pathogen if it's present. And I think the approach that they promote here in breaking down the pathway that's necessary for the detection of a disease is in itself very valuable. So this tool has been widely used on the terrestrial side, I think with considerable success. It's been little used on the aquatic side, so I'm really pleased to see that there's a presentation from Australia later today on the application of this tool. So, okay, so barriers to disease reporting. I put up this quotation here from a paper by Jenny Hutchinson and colleagues, because I think it highlights two really important features of surveillance systems. Firstly, that they are designed largely to meet the information needs of government. And secondly, often the disincentives to reporting outweigh the benefits that might be derived to those who are responsible for reporting, most importantly, farmers. So the kind of theme of this talk is a need to kind of shift the design of programs away from the informational needs of government, much more towards meeting the needs of farmers. I also want to commend this paper by Célia Bruguère, and it's not just because the senior author is on the stage. It's an excellent paper in which the reasons for underreporting are categorized as behavioral and technological. So on the technological side, it can merely be a lack of diagnostic tests, a lack of awareness by the farmers. On the behavioral side, as mentioned by Hutchinson, that there are disincentives potentially to reporting. There's a perceived fear of financial loss and potentially a lack of trust. So this is a newspaper article published in Ghana at the beginning of the year. So I'll just put the most important sentence at the bottom. So fishermen were disturbed about a trend in recent harvests. Over a month, fish had been developing severe skin infections characterized by red swollen lesions quickly turning into gaping wounds. So I'm guessing that some of you in the audience might have already reached a presumptive diagnosis. The interesting thing in terms of early detection was that the authorities were unaware of this problem until the newspaper article had been published. So clearly the fishermen were not keeping this a secret. They were concerned. They had reached out. The news of this problem had come to the attention of journalists. But the fishermen hadn't thought to seek help from either the local fishery officer or the Department of Veterinary Services. So I think it's important to try and understand why that is. I think, doing these post hoc investigations helps us understand why our early detection system is failing. And it could well be just because they didn't perceive that government was in a position to provide them any help, or it could just be a complete lack of awareness of what government services were available. Oh, and I should mention that, post this newspaper article, investigation was undertaken, and yes, Phanomyces invadans was detected as reported to WOA. So I think Melba showed a map of the distribution of EUS, and I think there needs to be another dot added a little bit further north on the African continent. Okay, so how can early detection be improved? So I think we need to make sure that the farmer is placed at the center of the early detection system, that reporting leads to investigation, that there's, as you say, diagnostic laboratory detection, and that importantly results, advice, and treatment are all generated, which are passed back to the farmer as benefits. And I would argue this creates a virtuous circle insofar as reporting results in benefits, results in more reporting. But of course, this does rely on trust, that reporting will result in investigation, diagnostics, and benefits. And in that system, I would argue that the informational needs of the competent authority will be met almost as a byproduct of a system that focuses on the needs of the farmer. Okay, so I'm going to move on, and that was, if you like, kind of back to basics. And I'm just checking on time. And then to move on and discuss a little bit more about the application of new technologies. And then putting this talk together, I realized that this was quite a large subject. So I'm having to restrict myself to 3 approaches to data acquisition. So mobile phone technology, molecular diagnostics, and remote sensing. And then I'll talk a little bit about the role of AI to bring together data and generation of information. So I would argue that the first priority for the application of new technologies, and I know mobile phones not exactly a new technology these days, is actually in support of farmer reporting. That must remain central. And there are some really good examples from Indonesia, work again by by Angus Cameron and others, where they have used text reporting to really improve the syndromic surveillance systems in Indonesia in terrestrial livestock. And I would argue that there are lessons that we can learn from the terrestrial side. And it's really great to see that in India you have an application, and in looking to that, it was clear that not only was the application there and supporting farmers, making it easy for farmers to report disease, elevated mortality, there was also built into that system feedback to the farmers. Clearly, diagnostics is crucially important. In developing countries, there are clearly constraints in terms of the availability of diagnostic capacity in purpose-built laboratories. There are often problems with transport and cold chain and the lack of expensive equipment. And to a significant extent, I would argue that these issues can all be addressed by the development of pond-side technology, using field-compatible molecular methods. And if those methods are using environmental samples, then that is an added advantage. And I'm not talking much about detection in wild populations, but clearly use of eDNA for surveillance of disease in wild populations is potentially a game changer. It's often actually, of course, finding and catching wild populations is a huge cost and constraint to surveillance. But most importantly, pond-side diagnostics allows rapid decision maker, be that by the farmer in terms of decisions around early harvest or treatment, or the competent authority in terms of speeding up implementation of measures to prevent the spread of diseases. And as already discussed, the time component is critical in ensuring a rapid and effective response. Okay, so remote sensing, and I should say at this point that CFAS have been quite heavily involved in development of what was referred to as smart buoy technology. So these are autonomous monitoring systems which generate high-frequency measures of a range of parameters with onboard computing and data transmission by satellite. So they've been quite widely applied in the marine environment. I would argue that they have considerable application around marine aquaculture sites. The parameters I've mentioned there, water temperature, dissolved oxygen, algal blooms, are all important risk factors for development of disease. So clearly, this information on a continuous basis potentially provides early warning for farmers about the risk of disease, allowing them potentially to take actions to mitigate those diseases, be that changes in stocking levels or preventative treatments. At a national level, again, this information is of use to the competent authority in the design of risk-based surveillance systems. So I'm focusing very much on passive surveillance, on reporting by farmers, but there clearly are instances when that system needs to be supplemented by active surveillance. And risk-based surveillance is, I think has been discussed earlier in the week, a cost-effective and efficient way of focusing resources. And this information is a way of parameterizing those risk-based surveillance systems. So these methods, I think, are starting to be applied in large inland water bodies, but I think there's a lot of potential, further potential for the application of these remote sensing to support aquatic animal health management. Okay, so just a bit of work that we did some years ago. So sea surface temperature data on a daily basis is quite widely available, but certainly in the UK, we don't have data on freshwater temperatures. But as we know, for many pathogens, temperature ranges are really important for establishment and expression of diseases, including koi herpes virus and spring viremia of carp, which are of interest to us in the UK, as well as I'm sure to many of you. So we took what was available, the air temperature data, gridded across the country, and used some available data sets to extrapolate daily water temperature data. And this allowed us to produce this, if you like, this heat map of where in the country establishment and expression of these pathogens was most likely to occur. And again, these information overlaid with population data allows us to identify areas which should be the focus of risk-based surveillance programs. Okay, so I need to talk a little bit about denominators. Obviously, for surveillance programs, the information on the location and type of farm is crucial to planning surveillance and disease control programs, but it's often lacking in many countries, and I think India may be an example of that. Aerial and satellite imaging, it has been effectively used to not just locate, but also categorize aquaculture businesses and estimate production. There's really good work demonstrating this in the Mediterranean. So I would argue that, you know, the use of this technology potentially overcomes some of our data constraints that we currently face. Okay, so I am coming somewhat to the end, so I wanted to try and tie some of these technologies together. So we've seen how you know, smart buoy technology can monitor important risk factor parameters. We've seen there are developments in DNA analysis, in microbial analysis, and the miniaturization of that technology. It's not a big stretch, I don't think, to see the combination of these technologies, which would allow, I think, both potentially early detection of diseases for specific assays, but also monitoring of changes in, let's say, the pond or the sea microbiome, which might act as important early indicators for disease emergence. So, going forward, we potentially are going to be faced with a situation where we have multiple sources of data, you know, not just, you know, SMS text messaging or from farmers, but continuous monitoring by smart buoys. I put up there automated mortality records. Haven't had time to get into that, but there's obviously considerable advances, particularly in salmonid systems, for detection of mortalities on an automatic basis, you know, and clearly crucial in terms of early detection of disease. But the challenge is going to be that it's going to potentially shift in surveillance systems away from data acquisition towards data analysis. And this is where artificial intelligence, particularly machine learning and large language models, have a role to integrate those data to produce information and support decision-making, rapid decision-making by both farmers and the competent authority. So a few conclusions. I'll just have that. So going forward, I would say that it is critical that we ensure that farmer reporting remains absolutely central to improving early detection systems for aquatic animal diseases. I think, first and foremost, the application of new technology needs to be to support farmer reporting, to make it easier and to make the data analysis rapid and effective. But that farmer reporting system needs to be within aquaculture health systems which are capable of disease investigation, generating support to farmers, and actually initiating measures to prevent and potentially eliminate pathogens. Of course, as there are the new technologies will also generate other sources of information. And I think they're going to have huge relevance to support surveillance systems where farmer reporting isn't adequate, so particularly in wild populations. So in advanced large-scale commercial aquaculture, the application of new technologies is already happening at pace. They are already benefiting from improved profitability and efficiency as a result of those methods. I think that our challenge is to ensure that small-scale farmers in developing countries are also able to benefit from these new technologies. So in the human health field, as I'm sure many of you will be aware, there's this concept of global public health goods. So these are goods which might be surveillance systems, it might be the dissemination of information systems, disease control programs, disease elimination. So importantly, the benefits of these goods cross boundaries. I would argue that early detection of aquatic animal diseases meets the criteria to be a global animal health good. And primarily because the benefits of early detection cross boundaries. They're not just going to be felt within the country in which it's implemented. And I think if we consider and promote early detection of aquatic animal disease as a global animal health good, I think it strengthens our argument for investment in early detection by government, by international organizations, by trade partners. And I think that it's important that an element of that investment is on ensuring that small-scale farmers benefit from the development of these new technologies. So I want to finish with that thought, but I need to acknowledge funding and CFAS colleagues who have worked with me on this presentation, and the WOA Aquatic Animal Commission, who I have the privilege of working with on some of these issues around surveillance. So thank you very much for your attention.
So thank you very much for this excellent presentation. So when we read the aquatic code, of course it talks about early detection, but how to implement this, what are the technologies to use, what are the limitations, and how to apply these and integrate the data that you get from different sources. You have illustrated very nicely in this presentation. Thank you very much. Any questions or comments? Yeah.
You mentioned about the importance of early detection of any disease. Now, for a disease like white spot virus syndrome, the time taken from the infection to the disease outbreak is very, very short period. It could happen in few hours if the conditions are ideal. Let's say the temperatures are ideal, about 27, 28 degrees. The disease outbreak is very rapid. Even before we could actually, do a diagnostics and confirm, you start seeing mortalities in the ponds. So how do you overcome this? Because even after 30 years of this viral disease affecting shrimp farming, it continues to be devastating the shrimp industry worldwide. And more so in India, quite recently also we had very large outbreaks of this disease. So how you think this early detection is going to help in such a case?
Right. So I am by no means an expert in white spot, but I suppose, at a generic level, I suppose, if come back to the point I briefly touched on, is there research opportunities to actually attempt to monitor the microbiome of the ponds in which the shrimp are being farmed, to try and identify potential changes in the microbiome that might act as early indicators for white spot emergence? I suppose the only other argument is, whether there are sampling of the shrimp and development of diagnostic tests that looks for markers of even earlier markers of infection. But I'm straying considerably outside my area of expertise when in this area. But
because the window period for the infection to break out into a disease and mortality is very, very short with white spot syndrome virus, given the ideal conditions. So this is a big challenge, even for putting in practical biosecurity in farms. That's one point. The other one which I wanted to ask you was, you mentioned about remote sensing of the temperature, doing the air temperature and correlating with the pond water temperature. How is that done?
So remote sensing of temperature?
Yeah.
So so there are, technologies are available for to remotely detect at temperature and to have that information passed back via satellite transmission to be stored centrally. I think the challenge in terms of its application for small-scale farmers is the role of government in actually promoting those systems and making and passing that analysing the data and passing that information back to farmers. I think that's the main challenge.
That's not ambient temperature, but we're talking about pond water temperatures. If there is a correlation, but that could be a significant indicator of the potential outbreak of diseases.
Yeah, I don't think I'm completely following your question. I think the key thing is that pathogens have a temperature range over which when disease will express. So monitoring those parameters potentially give farmers a opportunity to forecast when disease might strike. I think that's the issue.
Thank you.
Thank you very much. Yeah, just maybe the last question.
Thank you. I think you talked about trust. And coming from an industrialized country, Norway, I am actually experiencing much more about the compliance and the misuse of compliance, if we can say that, and combining it with the early detection, because the farmers don't really want to know, because then it comes with restrictions. So how do we really work with the incentives there?
So I think if I've got you right, you're referring to potential disincentives to reporting because they're worried about the consequences of
actually not even trying to analyze because you don't want to know.
Sure. So obviously, if compensation is paid for measures taken by government to control listed diseases, that's clearly potentially acts to overcome some of these issues around disincentives. I mean, the issue of compensation is complex and there are potential disadvantages of that. Some people have promoted a kind of like compromise whereby the first people to report listed diseases get compensation but not the others. Other systems have attempted to link compensation to biosecurity measures. If you've got all your biosecurity measures in place, then you're eligible for compensation. So there are no easy answers. I suppose it's to do with awareness and education insofar as making and trying to encourage a kind of cooperation between farmers and a realization that the negative consequences will spread to other farms. So if there is better, good partnership and cohesion within an industry, I think that goes a long way to supporting it. I mean, the other argument, which I'm not sure has ever really been properly implemented, is insurance schemes, that industry-run insurance schemes to support the few. The basic dilemma is you're expecting in the control of a listed disease for hopefully the few farms that are affected to bear the cost, but the benefits are for the whole industry for eradication of a disease. That's a fundamental issue that you potentially overcome through government compensation, but is that a good use of public funds? Arguably the benefits are to the industry, and therefore it is industry that needs to be seeking solutions through insurance schemes. Sorry, that was a...
Sir, one brief.
Thank you very much.
Sir, one brief question.
Yeah, I think many of you have questions. This is a very important topic. I know there are many hands, but we are running out of time.
One brief question, sir.
I will suggest that you please discuss with him during lunchtime. So, thank you. Thanks for your interest in this. And thanks, Edmund.
Epidemiology. This is the main course. Forget about the rest of the conference. That was just the hors d'oeuvre. That was just starters. Now we are here with the main course, epidemiology. And it's my pleasure to introduce Neeraj Sood, friend, colleague, somebody I've worked with for 12 years. Neeraj brings to his role as the coordinator or the consortium principal investigator of the National Surveillance Program. He brings three things to it. One is his integrity, one is his energy, and the other is his humility. He's a veterinarian, graduated from the Punjab Haryana University. He did his MSc there, pathology, PhD there as well. He was recognized some years ago as with the award of the best fisheries scientist, and recently he won an award for the, let me read this out, e-governance in aquatic animal disease management surveillance, but particularly related to citizen-centric sciences. Neeraj, it's my honor and pleasure to introduce you.
Very good afternoon, respected chairs and distinguished delegates. So, I'll be making a brief presentation on the National Surveillance Program for Aquatic Animal Diseases, and this program, it was initiated in 2013, and at present it is funded under the Pradhan Mantri Matsya Sampada Yojana by Department of Fisheries, Government of India, through National Fisheries Development Board. And this program, it is coordinated by our institute, that is ICR, National Bureau of Fish Genetic Resources, and our DDG, Dr. J.K. Jana, is the coordinator of this program. As Dr. Peeler was telling, that diseases are considered to be one of the major constraints to the growth of aquaculture. And there are several reports of economic losses due to aquatic animal diseases. In India, recently, Central Institute of Brackish-water Aquaculture, they had estimated the economic losses to a tune of about 2.5 billion in the aquaculture in the year 2021-22. And previously, the annual losses to the shrimp culture, they were estimated to be around $1 billion. So, considering the impact of aquatic animal diseases in the Department of Fisheries, Government of India, they had initiated a program, that is the National Surveillance Program for Aquatic Animal Diseases. This program was initiated in 2013, and the phase 1 of the program was from 2013 to 2022. The program was initiated in 14 states of aquaculture importance, and subsequently the program was extended to 19 states. And the major emphasis of the program has been to strengthen the passive disease surveillance and undertake the need-based surveillance. So we have the ICR Fishery Research Institutes and College of Fisheries, which were primarily involved in the surveillance program, besides some of the private colleges, as well as Rajiv Gandhi Center for Aquaculture, they were involved in the phase 1. And at the beginning of the program, we have developed a diagnostic manual for aquatic animal diseases of national concern, which includes the WHO recommended protocols for the WHO listed diseases, and some of the published protocols for other diseases of national concern. And we are really thankful to the World Organization for Animal Health for giving us permission to reproduce some of the protocols. And at the beginning of the program, our institute had collected the positive controls for the WHO listed diseases, and we are continuously upgrading this capability for detection of the new emerging diseases. And we are really thankful to all the world-listed WUHA referral laboratories, CSIRO, Norwegian Veterinary Institute, and Dr. Sonal Patel in particular, who have been kind enough to share the positive control. And I would like to mention here that network of aquaculture centers in Asia Pacific has been involved in the program right from the beginning. Dr. C.V. Mohan as the Research and Development Manager of the NACA, and subsequently Dr. Eduardo Lino, they have been involved in the program. And at the beginning of the program, we had some orientation training program involving NACA as well as Professor Kenton to sensitize the project investigator about the concept of the disease surveillance. So in the phase 2 of the program has been operational since 2022, and it is going to end in 2026. And this program was initiated when Dr. Kuldeep sir was the director of ICR and FGR, and he has been very instrumental in getting the grant. And in the phase 2, we have expanded to pan-India, and we are trying to involve the State Fisheries Department because of their contacts with the fishermen at the grassroots levels. And besides, we have also included Marine Products Export Development Authority, because they have been collecting a lot of samples for PHT as well as National Residue Control Program. So in this program, again, we have all the ICF Fisheries Research Institute, College of Fisheries in different states, and then we have National Fisheries Development Board, RGCA, as well as MPDA in the surveillance program. Besides, we have 3 referral laboratories at Central Institute of Freshwater Aquaculture, Central Institute of Fisheries Education, as well as Central Institute of Brackish-Water Aquaculture. So for strengthening of the passive disease surveillance, all the centers, they conduct awareness meetings, mass awareness meetings involving the State Fisheries Department to sensitize the farmers about the ongoing surveillance program. Besides, during these awareness meetings, we share the awareness literature, which is generally in the regional languages, and we also share the contact number of the project investigators whom the farmers they can contact in case of emergency. Besides, to strengthen the diagnostic capability of the State Fisheries Department. Officers of the State Fisheries Department, all the centers, they have been organizing training programs. So as Dr. Peeler was showing, that under the program in the phase 2, we have developed a report fish disease app. This is an Android-based app, and recently we have also developed an iOS version of this app. And this app, it is available in Hindi, English, and 11 regional languages. So the farmers, they can download the app, and then they can submit reports of any disease cases on the farms. This, I'd like to mention. So if you see, this has three panels. This report fish disease has 3 panels. One is the farmer dashboard, which is available on the Google Play Store. The farmers can download. Then they can enter some information. Mostly they have to just enter the information in the form of tick boxes. And then they can upload the photographs of the diseased aquatic animals. So based on the presumptive diagnosis, whenever the farmers, they submit the report, then it is shared with the officers of the State Fisheries Department and NASPAT collaborating centers in real time. And based on the presumptive diagnosis, some of the management measures can be suggested. However, in case it is felt that this case needs to be investigated, then our centers, they go and collect the samples, or we request the farmers to bring the samples for disease investigation. And at the coordinating institute, we can just monitor the progression, the action taken in different, regarding the disease cases reported by the farmers. And we are thankful to all our collaborating centers for their translation of the app in regional languages. So whenever the farmers, they report any disease outbreaks, we use different levels of diagnosis. I know Dr. Melba and FPO, they have been long emphasizing using level 1, level 2, and level 3 diagnosis. So whenever a disease case is reported, we check the water quality parameters, and then also we make level 1 diagnosis based on the clinical signs and gross lesions. And in many cases, level 1 diagnosis is adequate to suggest some management measures. And if required, then we go in for level 2 diagnosis, which includes wet mouth examination, and it provides very useful information because most of the disease outbreaks, they start at the mucosal surfaces. In addition, we use histopathology, bacterial isolation or fungal isolation, and then we also use level 3 diagnostic techniques, including virus isolation, electron microscopy, and molecular techniques for confirmation of the pathogen. So following investigation of the disease cases, we suggest some management measures which help reduce the economic losses due to the diseases. So the strengthening of the passive disease surveillance, it can be evidenced by first-time reports of several new diseases from the country for the first time. These include infection with the cyprian herpes virus 2, Coy sleepy disease, then tilapia lake virus disease, red cyprian iridovirus infection. Similarly, snakehead rhabdovirus infection, bacillary necrosis of fungus, which is caused by Advertila ictulari, tilapia parvovirus infection, as well as epitheliocyst in case of finfish. And in case of shellfishes, a number of new diseases like infectious myonecrosis, mudcrab virus infection, then infection with shrimp virus 8, Macrobrachium rosenbergii golda virus, as well as infection with E. cytonucleospora hepatopeniae. They have been detected under the surveillance program for the first time. So with the initiation, after the initiation of this implementation of this national surveillance program, a number of new diseases they have been reported during the last 12 years. So we have also evolved a mechanism for first-time reporting of a new disease. Whenever any of the centers comes across a new disease, the samples they are shared with 2 national referral laboratories, and following conformity in the results, a compiled report is prepared and submitted to the competent authority, that is the Department of Fisheries, Government of India. And in case of WUHA listed disease, the information, it is shared with the World Organization for Animal Health within the 24 hours, and at the same time, the competent authorities sends alerts and advices to the State Fisheries Department and other stockholders. And subsequently, the disease is reported included in the quarterly aquatic animal disease reports being submitted to the network of aquaculture centers in Asia Pacific as well as OAE. And here I would like to mention that our country has been very regular in submitting the quarterly aquatic animal disease reports, and we have been very transparent in disease reporting, despite some of the trade restrictions being imposed by some of the countries who are not doing any disease reporting. So I'll just like to give a few examples here. So in 2017, there were some mortalities in tilapia in West Bengal as well as in Kerala. Then one of the centers, that is Inland Fishery Research Institute, they detected tilapia virus in the samples, and simultaneously the PAGR center where Dr. Raja Swaminathan is there, so he also detected tilapia virus. And then subsequently we did histopathology, we used virus isolation, bioassay, and following confirmation a report was submitted to the competent authority. Again, I would like to emphasize that for first-time confirmation of the disease, we use multiple techniques for confirmation. And then we submit the report to the competent authority, which then sends alerts and advisories to the State Fisheries Department. And many of the State Fisheries Department, they have been sending these reports to the block level officers. Another recent example is the bacillary necrosis of fungus, and my colleague, Dr. Chandrabhushan, he has a poster presentation in the same session. So there were some mortalities in pungation around hypothalamus, and in the wet mount examination, we did not see any significant parasites. When we did the necropsy, we could see some white foci in the kidney, which was suggestive of bacillary necrosis of fungus. So subsequently, we detected the Advertila ictulari using species-specific primers, then the bacteria, and then confirmed using amplification and sequencing of some of the 16S ribosomal RNA gene as well as gyrase B gene. Then we also did some histopathology, and subsequently, using the culture, we did bioassay, and we could reproduce the typical disease, and we could re-isolate the bacterium, thereby fulfilling the Koch's postulates. So it's not only that we are just confirming the presence of the disease. We have been able to negate the occurrence of certain diseases from our country. In 2013, there were some reports of acute hepatopancreatic necrosis disease from the country. So subsequently, an emergency response team was constituted, which was led by Director, Central Institute of Brackish-Water Aquaculture, and this team undertook the targeted active surveillance, and none of the samples, they were found to be positive for Vibrio parahemolyticus AHPNG strains, and subsequently also we have been regularly doing screening for this pathogen, and till date, we have not come across any of the positive samples, and we still feel that we still have the evidence to show that our country is free from this deadly disease. So again, whenever all the centers, they have been investigating the disease cases, and based on the information collated, we find that in case of finfish, the parasitic diseases, namely the argulosis, lerneosis, dactyliogyrosis, they are the major diseases. Similarly, motile aluminate septicemia, then oomycete diseases like saprolagniasis and infection with Aphanomyces invadens, and some of the viral diseases like infection with infectious spleen and kidney necrosis virus, tilapia lake virus, tilapia parvovirus, and some of the other viral diseases that continue to be there in the finfish culture. Similarly, in case of crustaceans, hepatopancreatic microsporidiosis is one of the leading causes of losses, and this is followed by white spot syndrome, white spot disease, and in case of mollusks, the major diseases, they include infection with wool-listed Parkinson's olsoni, and then infection with the Parkinson's baihiensis. So all the information generated under the active surveillance as well as passive surveillance is being entered into a national database on aquatic animal diseases, which is maintained at ICR, National Bureau of Fish Genetic Resources, and this database was prepared in collaboration with National Institute of Veterinary Epidemiology and Disease Informatics. And in the beginning, we had designed some uniform format for collecting the information from the farm for the disease cases as well as for the active surveillance. So using the database, we can prepare the distribution of maps of the major diseases, and we can monitor the changes in distribution of the pathogens over time or in different geographies. Still, we need to have probably comprehensive information about the diseases to have meaningful information. Under the program, we have been organizing a number of international events like we have been involving some of the international organizations like a network of aquaculture centers in Asia Pacific, and then we have also organized schools on aquatic animal epidemiology, where Professor Kenton Morgan Lloyd has been kind enough to conduct 3 epidemiology schools for the project investigators as well as for the research scholars. And even after this symposium, Professor Kenton Morgan Lloyd will be conducting 2 epidemiology schools, one at Central Institute of Brackish-water Aquaculture and subsequently at the National Bureau of Fish Genetic Resources. And we are thankful to Professor Kenton for organizing all these schools free of cost. So coming to the concluding slide, I would like to mention that with the implementation of surveillance program, we have been able to create a strong network of diagnostic laboratories across the country, and there has been significant enhancement in our disease diagnosis. Then we have evolved mechanisms for first-time reporting of the new diseases and notifying the diseases to the World Organization for Animal Health. And most importantly, we have been providing scientific advice to the farmers following disease investigation. So recently, our lab has been recognized as a world referral laboratory for infection with Aphanomyces invadens, and we are also associate partner lab for World WUHA, collaborating center for emerging aquatic animal diseases, and we are thankful to Dr. Kelly for involving us in this as an associate partner lab. And in the end, I would like to thank the convener and the organizing team of the DAA 12 for giving us an opportunity to make the presentation, and we are thankful to Department of Fisheries and Government of India for funding this program through National Fisheries Development Board. We are thankful to the Indian Council of Agriculture Research, State Fisheries Department, and Team NASPAD. So this is the Team NASPAD. Thank you.
Thanks, Neeraj. Questions? Can I ask also that if you ask a question and you're not satisfied with the answer, you discuss it over lunch? So, any questions?
May?
Yes?
Yeah. I'm Dr. Ravi Shankar from ICRC. I'm an economist. Actually, missed the point. The project data helped our insurers to come out with the product for shrimp crop insurance. We have 5 products, out of which 4 are covering disease loss. And one more thing, request is, you have to reduce the drudgery of data input entry by farmers to increase the passive surveillance, because farmers are not willing to have a lot of clerical work like typing name, age, education and all. So now we have tried a small experiment with the engineering students of 3rd year. They came out with the Python code within 3 days. Just you talk, it makes the entry of name, like other voice recognition of data entry, and OCR reading of documents. If a lab report is there, just to show to your mobile phone, it reads the document and gives the input to your database. And reducing the file size or changing the format as per your requirement. Everything can be automated so that drudgery on the farmer's side data entry will be reduced. That is a simple request. And actual insurance company, they need data up to the last crop. It is not the previous time gap they are not willing to accept. Till last crop, they need to adjust the premium rate so that neither they fall into trap nor farmers are burdened with more premium. So thanks a lot for giving the data support for business, aquaculture insurance business also. Thank you very much.
Thank you. I'm going to take Melba first. She had her hand up first. I'll come back to you, Shankar. How many microphones do we have? Do we have more than one?
Thank you very much, Kenton.
There's 2. Can we get the next
Dr.
one? Dr.
Niraj, thank you very much for your excellent presentation. My question, and congratulations on the phase 2 of the surveillance program. My question is, hello, can you hear me? Okay, so my question is in your reporting flow. So there was an arrow, presumably I think you are reporting back to the stakeholders or the farmers, right? And you have mentioned that you have included about 45,000 farmers participated in this surveillance. Am I correct? Yeah. So 2 questions. I would like to know what sort of feedback information you are giving back to these 45K farmers who participated in your surveillance, and what do you think is the buy-in from the farmers to actively participate? Do they have a good understanding of the benefits of participating and the benefits of reporting? Is it clear?
So, yeah,
2 questions.
actually, for the awareness, during the awareness program, we sensitize them with the ongoing surveillance program, the major diseases and their management measures that they need to undertake. And then we also tell how to bring the samples for investigation, because many times what happens, they bring the dead fish or shrimps for disease investigation. We will tell them how to bring, whether to, they need to bring the samples in live condition or on ice. The moribund shrimps or fish they can collect on ice and then they can bring. So this kind of information we are sharing during the awareness programs. So
I mean feedback. Once you have collected the samples and analyzed, so you have
that's a lunchtime. That's a lunchtime conversation.
So we are sending the reports. So we share the information and suggest the management measures for that. We share the disease reports, disease investigation reports, and suggest the management measures. Yeah.
Hi, sir.
Shankar, I'm trusting you to be really short. Short question.
Yeah. Dr. Neeraj, at the moment, ICR and SAU are doing this job, network, and actually in the veterinary department, the department is doing the surveillance. When it is better the department does. When exactly this will be transferred to the departments of the various states?
Actually, sir, in phase 2, we have been trying a lot to involve the state fisheries departments. So we have been, about 9 to 10 state fisheries departments. We have already released some funds. We are conducting training programs. We have again returned to them for training the personals. But we need more involvement of the state fisheries departments to make this program successful, because reporting they can have a very significant role in reporting of the disease cases and transport of the samples, at least, to the nearby collaborating centers. So we have been trying a lot, and we hope that with this one, after some time, there will be more involvement of the State Fisheries Department, similar to that in animal husbandry sector.
Sir, this is Karthik from Aqua Exchange. Does NASPAD have any plans to do surveillance for the hatcheries, fish and shrimp hatcheries? Is my question number one.
So actually hatcheries, particularly shrimp hatcheries, they have their own laboratories. So they are giving they are testing the samples and giving the reports, I think, before they are sending this one to farmers, selling this one seed to the farmers. And if some of the hatcheries they are interested, then they can contact the nearest collaborating centers for the screening of the post-larvae.
And my question number two is...
No, sorry. No, number two. You
last
have to
one,
do that.
last one.
No, I'm sorry.
It's very important.
No, number two. I'm very sorry. We are now, because the students have prepared their posters, they've spent a lot of time doing it. We should give them the respect of examining the posters. I'm sorry, I don't like to stop questions, but you can ask a question during the lunch break. I'm very sorry to do that. We're running about half an hour late. It's very important, I think, that first of all, we thank Neeraj for an excellent presentation. And I would suggest we come back at 2:30.